Hi, I am Dr. Arsalan and today we are going to discuss about the MCQs related to the viral infections, especially AIDS. The first MCQ is HIV AIDS was first recognized in dash in dash. So he is asking about the viral infection AIDS, acquired immunodeficiency syndrome, which is caused by the virus human immunodeficiency virus. So I am going to write separations human immunodeficiency virus and the disease acquired immunodeficiency syndrome. So this is basically the causative agent and this is basically the disease. So this is the difference between HIV and AIDS. So he's asking about the country of origin or recognition and year of recognition. So AIDS was first recognized in the options given are USA, India, Africa, and Brazil. So the correct option is USA. The first blank will be filled with USA. As far as year is concerned, so AIDS was recognized in the options given are 1881, 1886, 1981, and 1986. So AIDS was recognized in 1981. So by completing your answer, we can say that HIV/AIDS was first recognized or reported in USA in 1981. Next question is the enzyme involved in the pathogenesis of HIV is. The questionnaire is asking about the enzyme which is required for pathogenesis or disease onset of HIV. The options given are RNA polymerase, DNA polymerase, reverse transcriptase, and protease. So we should be aware of the functions of these given enzymes. RNA polymerase enzyme is required for the process of transcription for the conversion of DNA into RNA. DNA polymerase is required for DNA replication, that is, making copies of DNA or synthesis of DNA. Reverse transcriptase enzyme, it is required for conversion of RNA into DNA, while protease is required for splitting of proteins or polypeptide chains. So among all these options, the most appropriate and correct option here is reverse transcriptase because the HIV virus, it is retrovirus and has genome comprising RNA. So for onset of the disease, HIV must convert its genome RNA into DNA, and for this purpose enzyme reverse transcriptase is required. So the correct option is reverse transcriptase. This is the enzyme required by this HIV virus to convert its genome of RNA into DNA. Then it will transcribe the DNA and translate into proteins forming the progeny viruses. Important pathogenic aspect of the HIV is based upon the reverse transcriptase enzyme. Next question is HIV binds to the receptor CD4 proteins located on. So it's very interesting question. It involves the basic receptors to which the HIV virus binds. This is called CD4 receptors. So the questionnaire is asking that the CD4 receptors, these are basically antigenic proteins, are located on which type of the cells. Given options are cytotoxic T cells, helper T cells, and suppressor T cells. These three options are the types of T lymphocytes. Cytotoxic T cells kill the antigen, helper T cells helps in the recognition of the antigen, and suppressor T cells reduce the overactivity of T lymphocytes. Next is B cells or B lymphocytes. So B cells or B lymphocytes do not have any concern related to the HIV infection. So this option will be excluded. Now the CD4 receptors are located on helper T cells. These are not present on suppressor or cytotoxic T cells. So these are located on the helper T cells. The correct helper T cells have antigen proteins called CD4 present on their surface to which the HIV virus attaches or binds and initiate the infection process. Next question is there are dash stages of HIV infection. Options given are 1, 2, 3, 4. The question appears very simple, but it is of utmost importance. It's very much technical question asking about the stages of HIV infection. So the correct option is 3 because there are 3 stages of HIV infection. First one is acute, second is chronic stage, and third is diseased or clinical stage. Acute stage involves initial infection stage in which it may take up to 2 to 4 weeks of time. The virus initiates its pathogenesis in the body. Stage early flu-like symptoms appear in some patients. Flu-like signs are the characteristic feature of this infection stage. Second stage is called chronic stage. It lasts for years and it is latent phase in which there are no prominent signs. The virus is undergoing multiplication and the disease is in progressive stage without showing any signs and symptoms. The last stage is diseased or clinical stage in which the prominent signs, the clinical manifestation of AIDS occur, and this is also called symptomatic phase. At this stage there is no cure for the disease. Clinical manifestation has occurred, the immune system of the individual has been damaged. So this is the last and advanced stage of HIV infection. Fifth question is HIV is the mutant form derived from dash through mutation. So he is asking about the origin of HIV virus, that it has mutated from that kind of virus. The given options are SIV, simian immunodeficiency virus. Next option is Congo virus. Third option is chimpanzee HIV, and fourth is Ebola virus. Simian immunodeficiency virus, it causes AIDS in chimpanzee. Second is Congo virus, it causes Crimean Congo hemorrhagic fever. Crimean Congo hemorrhagic fever, which is tick-borne disease. It is transmitted through ticks. Chimpanzee HIV, as we have stated that simian immunodeficiency virus causes AIDS in chimpanzee. There is no HIV virus in chimpanzee. Chimpanzee has infective SIV, simian immunodeficiency virus. So this is incorrect option. And next is Ebola virus, which causes hemorrhagic fever in human beings. So among these options, the most appropriate option is SIV. The HIV virus has got mutated from the simian immunodeficiency virus, which causes AIDS in chimpanzee and non-human primates. To convey solidarity with the AIDS patients, general public designated dash as World AIDS Day. So this question belongs to general knowledge, that World AIDS Day is celebrated on. Given options are 1st December, 25th December, 6th June, and 6th January. So the correct option is 1st December. 1st December is designated and ranked as World AIDS Day to convey solidarity of the general public with the AIDS patients and to create awareness in the general community. And they have a symbol of red ribbon to show the solidarity with the AIDS patients. This question is related to the genome of the HIV virus. So the genome of HIV is made up of dash. The given options are single-stranded RNA molecule, double-stranded RNA, single-stranded DNA, or double-stranded DNA. The correct option is single-stranded RNA molecules. If we elaborate it, so the genome of HIV virus is made up of two molecules of positive sense single-stranded RNA. So the genome is basically single-stranded RNA which is of positive sense, and there are two molecules of RNA. Next question is trimeric proteins embedded in the envelope of HIV are glycoproteins 120, glycoprotein 41, and dash. So this question is itself informative. that the envelope of HIV is made up of trimeric proteins that are embedded in the envelope. HIV is enveloped virus, so it reveals that HIV is basically enveloped virus, and its envelope is made up of three kinds of proteins called trimeric proteins. These are glycoprotein 120, 41, and the next option is P24, P17, glycoprotein, and ENV. So option is very clear and obvious in this, that P24, this is matrix protein. It means that the matrix of the HIV virus is constituted by P24 and P17 proteins. These are matrix proteins. GP is general term glycoprotein, which are already stated there. The correct option is ENV, enveloped protein. So these trimeric proteins, these constitute the envelope of HIV virus. These are glycoprotein 120, 41, and ENV. Next question is, the attachment receptor of HIV is CD4 molecule located on helper T cells, macrophages, and dash cells. So again, in this question I try to convey some basic information of the HIV receptors, that HIV virus has receptor molecules of CD4 present on helper T cells. So these are the helper T lymphocytes on which CD4 receptors are present, and the HIV virus, it binds to the CD4 receptors located on T cells. Also, these receptors are present on macrophages. And third option he is asking about. These options are neuroglial cells, microglial cells, B cells, and NK cells. Neuroglial cells are basically cells of nervous system. These are non-neuronal cells which do not transmit any impulse, rather they support the neurons to transmit information. So this is a relevant option. Next is microglial cells. These are basically macrophages located in central nervous system. Macrophages in central nervous system are called microglial cells. This is the correct option, the desired option. The microglial cells, the macrophages in CNS also comprises CD4 receptors to which the HIV can bind. Next are B lymphocytes. B lymphocytes have no concern with the HIV infection. And last option is natural killer cell. It is a kind of T lymphocytes. Natural killer cells are a kind of T lymphocytes. So the correct option is macrophages in CNS are called microglial cells. Next question is, dash group antigen is required for core protein. He is asking about the structure of core in which the matrix and RNA molecules are present in the HIV virus. This core is made up of proteins. The given options are polymerase, GAG, ENV, or reverse transcriptase. So all of the proteins are very much familiar to us, and we have discussed in detail the functions and types of these proteins in our lecture. Polymerases are required for DNA replication or transcription process. GAG protein, group specific antigen. GAG protein means group specific antigen. So these are the basically structural proteins which form or synthesize the core of HIV virus. This is the correct option. Other is ENV. We have discussed that it is required in the formation of envelope of the HIV, and reverse transcriptase is required for conversion of RNA of HIV into DNA molecule in its life cycle. So the last question is, HIV uses dash as factory because viral DNA incorporates into these cells and produce viral particles. So the cells used by the HIV for incorporation of its DNA to take over the biosynthetic machinery of that cell and produce its own progeny, its own proviruses. These cells are, the options given are T cells are T lymphocytes, B lymphocytes, macrophages, and CD4. T cells are infected by the HIV. B cells are related to the humoral immunity, are for synthesis of antibodies, have no concern with the HIV attachment or synthesis. The macrophages, and last is CD4 molecule. This is the receptor site of the HIV, so to which the HIV attaches for infection. The correct option here is macrophages. Macrophages are those type of cell in which the virus incorporates its DNA and synthesizes its progeny viruses inside the macrophages. So these are basically reservoir or host cell for the HIV to initiate and propagate its generation. The correct option here is macrophages. So hope this video was useful for you. Thanks for watching and good luck.